Good morning, everyone. And my name is Hee Kim, and I'm from South Korea. I am very nervous because I'm the first presenter in here. And actually, this is the first time in Japan. And yesterday I came here, and I'm very impressed. The Japanese is very, very, very kind and very, very gentle. And it was really good in the yesterday dinner. And thank you. It's such a great honor to be here. And first of all, Sapporo is the hometown of my favorite beer, and it was very pleasure. The Professor Ikeda and I met in ISE International Conference in 2023 in Taiwan, and this year we met again in Atlanta. In December, last August. I'm truly honored to be here, and would like to thank Professor Ikeda for kind invitation and hospitality. Let me introduce myself. I'm Professor Hee Kim, and I work at the Kyung University in South Korea. And my background is nursing and public health. And my research is focused on the environmental exposure, epidemiology, and information. My title of lab is Poison Lab. Poison Lab means protect women and infants from substance of hazard and nursing lab. And I work with my master and doctoral student. Today, I will be presenting on risk assessment of infant exposure to EDC, PFAS, and heavy metals by breastfeeding, what we need to know. Shall we start? The breastfeeding is widely recognized as the optimal source of nutrition for infants, providing essential nutrient and immune protection for healthy growth and development. However, human breast milk can also serve as a pathway for transferring environmental pollutants, including PFAS, heavy metal, and EDC, from mothers to infants. EDC in the maternal body can accumulate in mammary tissue through passive diffusion, protein-mediated transport, or the active transport. And these are secreted into breast milk by globular cells. These mechanisms allow EDC to be transferred to newborn during breastfeeding, contributing to early life chemical exposure. Since 2018, I have been measuring the concentration of EDC, PFAS, and heavy metals in breast milk from Korean mothers, and in collaboration with the Korean Food and Drug Safety Evaluation Institute, conducting risk assessment for infants exposed via breastfeeding. Today, I would like to discuss with you what we need to know based on the insight I have gained from this research. This is a data source. I utilized this sample result from healthy mothers in the Life Epic code. This code established since 2018 at Kyung University. The life code EPIC means longitudinal impact of fetal and early life, exposure to persistent and industrial chemicals. The life EPIC code is a small code developed to investigate the impact of environmental hazardous exposure during pregnancy on fetal and infant health. We analyze the data, highlight disclaimers, parabens, PFAS, and the heavy metals in breast milk. Live Epicot recruited healthy pregnant women without complication during the first trimester and collected data and four times point the first trimester and second trimester, third trimester after delivery, and when the child reached 3 years of age. Were collected at all 4 times, while the blood and breast milk and these times and 1 time, 1 each. In addition, assessment of growth rate, maternal depression, and the neurodevelopment were conducted. This is Richard. I think the contamination is shifting from the traditional pollutant to substitute chemicals. This is the recent research. In the US study, the human exposure to phthalate has decreased by over 90%, while the substitute DHTP and DINCI have increased. In the Hokkaido study, DHTP and DINCI metabolites were detected in 95 and 92% of samples. During the 6-year period, DHTP levels increased about 5-fold and DINCI level doubled. In the German HBM, exposure to traditional phthalate like DHTP has decreased, while the exposure to substitutes such as the DHTP and DINCI has increased. This pattern is the same in PFAS and bisphenol. In Korean human biomonitoring study, BPA exposure has decreased by 43%, while the PFAS exposure has increased 2.3-fold and PPF exposure increasing 20. In the Australian HBM study, PFOS and PFOA concentration have decreased, while the PFNA shows the increasing trend. In European HBM policy brief, combined exposure to PFH excess and PFNA has emerged as a dominant PFAS pattern. I wonder this pattern appears in the breast milk. So I reviewed the concentration of the PFAS and the EDC in breast milk in Korea. This is the phthalate concentration in human breast milk. The red bar is the Korean data, the blue bar is the another country data. The levels of NAHP, NNBP, and MIBP in breast milk from Korea are lower than in other countries. The Korean data shows this one. The Korean data shows the decreasing trend over time. But the levels of DHTP, DIN, GH, TOTM, and ATBC and the TXID, as you know, this substance is a phthalate substitute, and this one in breast milk were not only higher than those in other countries, but also higher than the concentration of other phthalate. The reason why that South Korea has stressed the regulation of phthalate since 2007. As a result of this strict control, the monitoring of human exposure has revealed a decrease in the detection of these traditional phthalate in human biology sample. Concurrently, there has been an increasing detection of alternative such as DINCI, TAMS, and ATBC. Importantly, this trend is also observed in breast milk sample. This is the bisphenols concentration in human breast milk. This is saying the red bar is the Korean data, the yellow bar is the Japan data.
The blue bar is the other country. It is shown the level of BPA in breast milk from Korea are lower than those in other countries, and the Korean data shows the decreasing trend. The BPA has also been banned in food container and the product for the infant and young children in South Korea. Recent monitoring data from CONEX and shows that the BPA level have decreased by 40%. In contrast, the level of the BPA substitute such as BPS and BPF increased. This one data. The levels of the left graph is the BPF, right is BPS. The levels of BPF in breast milk from Korea are higher than those in other countries. This one. This is the PFAS concentration in human breast milk, read by the Korean data. The Korean data shows that PFOA level remain relatively constant, but the PFAS level are decreasing over time. In South Korea, PFAS are designated as a restricted substance in 2013, PFOA in 2015, and PFHS in 2023. Considering the long half-life of PFAS, this data represents reasonable results. This is the PFHS and PFNA. Although data on substitute are limited, concentration of PFOA and PFOS in breast milk are decreasing, while the concentration of PFAS substitute, such as this one, are increasing in Korea. In summary, in Korea, the levels of the phthalated bisphenols and PFAS in breast milk have been lower than those reported in other countries, largely thanks to the effective regulatory measures. But this does not mean that contamination has disappeared. Rather, it has shifted from the traditional chemical to their substitute. For example, the levels of the DING and ATBC, which are phthalate alternatives, have been increasing. Similarly, BPF and BPS are rising as a replacement for BPA, and PFHSS and PFS are emerging in place of PFOS and PFOA. Considering the biologic characteristics of newborn, we should pay more attention to their exposure to substitute chemicals. The second thing we have to know, we need to know the risk assessment is environmental sensitivity that it varies depending on the international standard. This is the risk assessment to the bisphenol's exposure by breastfeeding. As you know, in 2023, EFSA lowered the TDI for BPA from the 4 kilogram body weight per day to 0.2 nanogram per kilogram body weight per day. We conducted a risk assessment of BPA by breastfeeding, and all samples, all samples were within the EPA and Korean TDI limit. The green bar is the EPA and TDI, red is the Korean TDI level. As you know, the all samples were within the EPA and Korean TDI limit, but when applying the EFSA new standard, the most of this one, the most of BPA EDI exceeded the TDI level. This highlights the importance of the regulatory standard. This is the risk assessment of PFAS exposure by breastfeeding. We calculated this one, the EDI for the PFAS in breast milk, and all of which are within the TDI level. This one. But in 2020, EFSA has set a combined threshold of 4.4 nanogram per kilogram body weight per week for PFOA, PFOS, PFHS and PFNA, reflecting the heightening the regulatory concerns of PFAS accumulation and potential health risk. In our data, some samples, this one, and some samples exceeded the EFSA TDI. In summary, the EDI of the character of bisphenol and PFAS through breastfeeding was generally below the TDI level established by international agencies such as the EPA and the EFSA, indicating an overall low risk. But when applying the recent revised, more strict TDI value for bisphenols and PFAS, most infants fall into the potential risk group. Of course, Korea has not adopted this threshold yet. Breast milk, but as we know, the breast milk is a unique biologic matrix. It serves as the valuable biomonitoring sample for assessing maternal health, while at the same time being a crucial source of the nutrient for newborns. Considering the unique characteristic of breast milk, I believe the stricter standard should be considered. Third, we also need to pay attention to arsenic exposure among heavy metals. This is the heavy metals concentration in human breast milk, lead, cadmium, the mercury, and arsenic. The red bar is the Korean data. The levels of the lead, cadmium, and mercury in breast milk from Korea are lower compared to other countries, but the levels of arsenic is this one. The arsenic is higher than other countries. This is the risk assessment of heavy metal exposure via breastfeeding, this one. The EDI for lead, mercury, and cadmium did not exceed the TDI level set by international and Korean standard, but this one, the arsenic exposure, the EDI of the arsenic exceeded the TDI level in one sample. This finding was also confirmed in the HQ result, this one. The arsenic main source of the exposure is rice, which is the staple food in Asian country. The high arsenic level in breast milk indicated that we needed to pay attention to the mother's diet during the breastfeeding. Conclusion, fertility breast milk are shifting from the traditional chemicals to their substitute. And second is the risk assessment criteria for breastfeeding should be strengthened. And third, we need to pay attention to arsenic exposure. There are many gaps in human milk research. I would like to, in the future, I would like to collaboration with the Hokkaido University. Thank you. This is the Kyung University photo. In the future, I would like to invite all of you. Thank you so much.